Hi, everybody. Oh, and this is being recorded. Got it. Everybody in person and everybody online. So it's my great pleasure to introduce our speaker today, Dr. Julia Zeitlinger. Oh, there's a little bit of an echo, but yeah, it's going to go away. So Julia is visiting us from the Stowers Institute of Medical Research, and she is also an associate professor at the University of Kansas. Julia got her PhD at Ember, and then she went to MIT for a postdoc with Richard Young, who you might know is one of the giants in the field of transcription and transcriptional gene regulation. So Julia's research has been very interdisciplinary. It sits at the intersection of transcription, developmental biology, and also genomics, and also computation. So her lab has been developing experimental methods, and you might know about this technology called chip nexus that can be used to look at very fine-grained, high-resolution transcription factor binding assays. And she's also been pushing on the computational side of things. So her research really focuses on one of the fundamental questions of gene regulation, that is how transcription factors figure out where to bind in the genome. And so she's been really a thought leader in really shaping the community in terms of thinking about both on the experimental and computational side of things as to how regulation happens at this level. And she's also been one of the early adopters of deep learning based methods to try to really get a more unbiased understanding of how gene regulation happens at this level. She's been really outstanding in terms of her accomplishment. She has many awards. Just to name a few, she got the NIH Director's Innovator Award, and she also is a Pew Scholar. She's a great mentor. Her trainees have gone on to become professors as well as gone into industry positions. And she's a really nice person to talk to. Every time I talk to her, she's very down to earth and super nice. So please join me in welcoming Julia to Wisconsin.
Well, thank you so much for the very nice introduction. I really enjoyed that. And I think you framed my talk so nicely. So thank you. Okay, so I'm going to present some unpublished work that we developed with the neural network approach that we're taking in the lab. But I'm going to give you a really big picture overview where I think the field is and could be going. I'm going to present you a little bit about the work that we did with neural networks and then where we're going to take that. So my lab, as Sushmita pointed out, is interested in understanding how DNA sequence controls development. So development is a very defined system. It is very reproducible. And you need to make these beautiful expression patterns in order to form all the parts of the body. And so that is, of course, a problem of transcriptional regulation. Oops, doesn't work. So in order to understand how sequence encodes gene regulation, we need to understand how transcription factor binds to so-called enhancer elements in the combinatorial fashion to activate genes. And this is a very complicated code. So this is a very well-known enhancer in Drosophila, and we know the transcription factors that are binding here. But if you look closely, these motifs are not exactly the same. They're actually not that easy to identify. And we also don't know what is it exactly about this arrangement that's important. Like, is it important, the space between, and how do they really activate exactly gene expression at the right time and the right place in the embryo? And luckily, though, this problem is no longer a sort of one enhancer at a time problem, but something that we can address with genomics technology. As Sushmita said, since my postdoc, I was really interested in understanding this problem from a more general perspective, not just for one specific system. And so in order to do this, we can leverage all the genomic data, in theory, to do this. And so now when you look at, we have a range of sequences, we can look where transcription factors bind in vitro very well. So we already have a collection of that. We can look where transcription factor bind in specific cells across the genome, such as ChIP-seq data, or we look at chromatin accessibility, which is also a good indicator. We can also look at things that are more correlating with the actual activity of the enhancers, which is histone modifications, for example, enhancer transcription by Pol2 also, or we can use reporter assays to measure whether that particular DNA element indeed drives expression. Now, on the other side, of course, we want to know what genes will be activated. So there is also, of course, the 3D organization of the nucleus. We also can measure transcription that happens, the nascent transcription. And of course, we have now single cell technology to look across many, many cell types and look at their gene expression programs. And I should say the cis-regulatory code is a hard problem because it is so cell type specific. Of course, we can also look at proteins. We can look at perturbations. So whether it's CRISPR now or synthetic libraries. So there's lots and lots of data available that really should allow us to ask the question, how do we put all of this together? But this is also the problem. And so you may encounter that when you read papers in the literature, genomics paper tend to be kind of on the descriptive side, or, so you wonder, like, how do we really extract this? And I think this sort of frustration is shared by many people. So this is just one quote that I think kind of captures that. Technology gives us the tools to analyze organisms at all scales, but we're drowning in a sea of data and thirsting for some theoretical framework with which to understand it. So this was Sidney Brenner, was in fact 10 years ago. And I would argue that we do have a theoretical framework, that I don't think we're missing something completely here. We just need to have a framework and tools to do this better. And I would argue this framework is pretty old, and that evolved in the 1950s, because already since the 1950s, we knew that DNA is really where all this information is encoded. So starting from Oswald Avery finding that really DNA was the transforming agent for a disease, not the protein. Of course, he wasn't believed at the time. It was only in the 50s when this was more accepted. But from then on, we knew how much information is encoded in DNA. And of course, the structure of DNA, I don't have to go into detail, makes it very clear that that there is a code that we can store and copy this information. And at the time, it was also clear that we can alter this information, like on fruit flies, of course, very old model system, and this information is inherited. Okay, so since the 50s, but I would argue that today we are in a similar time where DNA is, again, sort of in some ways a little bit neglected. And so we do know that there's lots of information encoded in the human genome that we cannot yet quite decode. So this is just an example from a GWAS study. So we have all these hits. And in most cases, we don't even know exactly which mutation is causative. And even if we do, we don't know the mechanisms, the cell types by which the trait that we're looking for is caused. We also have an incredible capability of sequencing DNA these days. I mean, it's getting like into $100 genomes. That's incredible. And yet we still cannot leverage fully that information. We also have the ability to edit DNA now with CRISPR technology exploding. And yet, we still don't know exactly what to do with that. So why is that? Well, to some extent, of course, it's historical. There's always it takes a little bit of a paradigm shift. And the last decades have been hugely successful, of course, in understanding the molecular basis of many, many cellular processes of diseases. So if you look at molecular genetics, biochemistry, structural studies, and now also perturbations are very, very precise, right? And so that's fun, and we're finding out a lot of things. But this is different in the sense that we can still do this with our human minds. We can look at a few components at a time and can make sense out of that. And DNA, unfortunately, is more complex than that. And I think this has been why it's been kind of resistant to, I mean, it's not that people haven't looked at DNA, but it's so complex that it seems sort of much harder to build a framework around that. So what we really need is technology also to change in order to go back to what's really written in DNA sequence. And as Sushmita mentioned, neural networks is kind of what we found to be really, really helpful with that. So just to give you an intuition, I mean, neural networks are everywhere. I'm sure the large language models are now all in the media. But even if you look at your phone already, it's very good at facial recognition. So you can just tag a person and it will give you all the picture, which is amazing, right? So why is that? So these neural networks are trained from the bottom up with large amount of data, but the data themselves can be fairly sparse, and that's an advantage. So this is the idea of a neural network with many layers, so it's deep learning, but it learns by, from the bottom up. So we're starting here initially with, just looking for edges. It detects edges, and it puts this information together in a higher order fashion. So we look for noses, eyes, and then put this together, information together as a face, and then taken together, it will make a prediction. So even though it's trained on faces, you can then easily train it on a particular person. So this is sort of the same idea with DNA sequence, that you look for sort of basic elements, but you can then put it together in very complex fashions, and it will learn so in an unbiased way. You don't have to tell us what to learn. It would simply learn the rules that are necessary to get the job done, and in this case, to learn genomics data. Now, this is quite different from what we're used to do. And I think that's kind of what often people stumble upon. It really is different. So traditionally, when we take a genomic data set, such as transcription factor binding, we take it apart. So we take the regions that say that are bound by a transcription factor, and then we ask, what are the patterns that we find there as motifs or co-occurrences of motifs? And so we're deriving an abstract representation of what we think is happening. The problem with that is that it's then very hard to go back. So it doesn't really make good predictions. Like in few regions, you may see that pattern, but in many regions you don't, and you don't really know why the transcription factor is bound there. So it has a poor predictive accuracy. Now, the complete opposite is true for deep learning. So I call that the inverted learning paradigm, because what you're doing is you're training, first of all, you're training on the entire, not the entire genome. So we're training it on regions, on larger regions, and we're letting the network learn whatever it needs to learn, in a combinatorial way in order to predict the data that we generated. And so we're achieving high predictive accuracy by definition. I mean, if we're giving it the right training set, of course, and we're testing on held out data, we can tell basically that the network learned the sequence rule from scratch in a very general way. And so that means we can look at an individual region and now say, okay, this is what the model was able to predict that region, and here's why. And this is really, really powerful. Now, of course, the problem with that is that it's just a prediction. It doesn't actually tell us what the rules are that it learned, because we didn't put it in. And that was good that we didn't put it in, but we need to now extract that. A neural network is sort of often called a black box model because, it learns just anything. And so we have to extract the rules from the network as a sort of secondary step. Now, of course, the easiest way to do that is simply use the neural network as an oracle, what it's called. So you basically mutating things, for example, or you give it something else that's still similar to your data set and asking, what happens if I do this? What happens if I mutate the motif, for example? And it'll make very accurate prediction. And that will tell you also what might happen if you mutate something, or it gives you also, if you do it in a systematic way, you can extract rules out of that too. So what we need to do is to have, first of all, high accuracy. We need to be confident that the model is good and that it really did learn biology. And then we need to go in and interpret. And there are already tools out there, but not everything is necessarily kind of served on the plate. Okay. So the question is, what can we do with that? So I've already told you, it's not just like in a better method of something that we did before. It is fundamentally different. And it took me a while to also see that. And I'm really excited about that because there's so much we can do with that. And I want you to think a little bit about that before I sort of, show you a lot of data. So what do we expect? Well, so you would expect that you get better motifs, right? And it learns these motifs from de novo, so we don't have to give it anything. And these should be really good motifs, right? They should be better. Okay. Well, now we also want to know, well, how do transcription factors interact with each other? So that should also be in there, right? You would expect. So we could ask, well, what does the distance between the motifs matter in their cooperativity? And not just that, but we don't just want to know yes or no. We want to have very precise quantitative rules by which we can now describe what's happening. Now, ideally, we want to link that to mechanisms. So I come from the biology side, from the transcription side. And so, of course, I want to be able to say, oh, okay, these proteins are interacting, or we want to explain what's really happening. And of course, initially, we were looking mostly at transcription factor binding, but of course, we want to get to activity, to target genes, right? And if we can do this well, we should also be able to redesign something. And this is sort of something really cool, like a synthetic biology approach. These networks are so accurate in the predictions. So now, in the past, it's been really hard to manipulate enhancers because usually once you manipulate, you destroy something and you have no idea what happened. But with neural networks, you should be able to do that. And ultimately, you would think that what we're learning is really anchored in biophysics, right? And for me, that's very hopeful because I know how complicated the cis-regulatory code is and how different it can be in different cell types. But fundamentally, they're all following the same laws of biophysics. So there's something in all cells that is probably similar. And I think we should be able to link that back to what's really happening. And that's exciting. So for once, for example, we might not only find motifs, but might be able to predict motif affinities. And I'm saying this, yes, because it actually does work. And Anshul has done a proof of principle. It's not out yet as a preprint, where they really, really showed that systematically. But we do see exactly that. So it's not just motifs, but it predicts the relative affinities really quite accurately. So we are, in fact, seeing these kind of things. Okay, so just to give you a little bit of a taste of the published work, what we did look at whether it matters, whether the distance, the arrangement of the motif matters for the cooperativity of the transcription factors. And this was also our original motivation. So we developed this technique called ChipNexus, which measures transcription factor binding in vivo at really high accuracy because we're digesting the DNA with an exonuclease. And so we're mapping exactly the stop base. And so you get these beautiful footprints that are stranded. And when we generated this data, it was clear to us that we couldn't really extract the information out of that. And so it was really in collaboration with Anshul's lab, where we're now using neural networks to predict these very high resolution data at base resolution. And this is something that, it came out of a necessity to interpret our data, but it turns out that this way of prediction is really, really good for almost any data set, because we're not just predicting something that's bound or not bound, so it's not a classification task, but we're predicting every base, and we're also predicting counts. So it is a very quantitative readout that we're asking the network to do. And I know from genetic screens that that's key, because the more quantitative your readout of a genetic screen is, also the better the results. And it's the same thing here. So this was the collaboration, and that took a lot of people, I should say, to develop even basic tools to allow this. But it was very exciting to happen. It was led by Giga Ausik, who was in Julian Gagneur's lab. So he really kind of developed BPNet when he visited Anshul's lab, and then working with me also in these sort of analysis of the data. So we started with mouse embryonic stem cells because we wanted a system that was really well studied, so we could sort of benchmark what we saw with the neural network. And so postdoc in my lab, Robin, generated beautiful data for OCT4, SOX2, and NANOG. And here you just see that it's, this is every region. So individual region, you see this very nice footprint. So the two strand, the red and the blue, or the resolution is also super high. We see higher specificity than ChIP-seq data because, for example, here we're looking at OCT4 at the SOX2 side, shouldn't really be there. ChIP-seq, you do see a signal, but ChIP-nexus is less. So we knew we have really nice data, but we really needed the neural networks to tell us what's in the data. So what do we see? So I should say classically, there's been sort of a big problem or kind of a debate in the field of how enhancers and how transcription factors work in a combination and whether the arrangement matters. So traditionally, at least back in the days, and this started from work back in the 90s, it's been sort of assumed that transcription factor bind in a highly cooperative way, perhaps like almost like a very sort of constrained way, so that they, almost like Lego bricks that sort of have to match. Although when you read the paper here from 2007, it's already clear that these protein-protein interactions are not that tight. They're actually much looser. But still, that original enhancer-zone model suggested that there has to be syntax. It has to matter if the proteins sort of interact with each other. On the other hand, there's also a model out there that still sometimes sounds like, well, it doesn't really matter as long as you have, so this would be, the billboard would be information display by each motif. And if you drive down the highway, you're getting this information display from different billboards, but the order doesn't really matter, right? As long as the information is there. Now, it's not that people haven't thought it might be in the middle, but how exactly was not clear. And the network was really giving us very clear rules. So this was nice to see. And they are both, there are two types we found. So one is what we call the strict syntax, where we do indeed see, and this is based on a previously existing structure, so this was known in this case, that there are two proteins, OCT4 and SOX2, that are binding cooperatively to a strictly spaced motif. So this makes sense that there was a strict space between these two motifs. But in many cases, we see something called a soft syntax. So the motifs are not strictly spaced, so and they do promote cooperativity, but it's in a distance-dependent manner. So even if you're a little bit further away, you're still getting a boost. But if you're closer, you're getting a stronger boost. There's stronger cooperativity. So what's displayed is sort of an enhancement of binding based on if the other transcription factor wasn't there. So we're seeing the sort of functions that two transcription factors have with each other. And noticeable, it's not always mutual. This is sort of based on thermodynamics. You would expect that it's mutual, but it's actually not. So in most cases, one transcription factor helps the other one more. And this is something curious. I can tell you now, we think it has a lot to do with chromatin. But I'm going to walk you through briefly of how this works. I mean, I already gave you the sort of the gist of it. So what we do is we train the data basically on the data that we already have. We train the network to predict something from sequence, and we're predicting the data that we have. And of course, we're testing on held data, so it's not like just memorizing the data, but it really has to learn the sequence combinations from scratch. So initially motif detection, but it also will combine it, and at the end generate the footprint. Once we have a good model, so here's just an example of this kind of predictions. It does really, really well. You can read up on that. But once we have the model, the key is then to go in and ask, how did it do that? And so the easiest way to do is sort of an attribution method where you trace back the signal to the input and give it weights. And the nice thing is when you do that, and this is just one region here, it gives you already weights, and the weights are clearly looking like motifs. The nice thing is you also see negative contribution. So that's not what you usually see in the frequency matrix, but it will say like, oh, I don't like that T or that G in that position. Because you have to remember, this all comes from individual regions. So when you now see individual regions, and of course it's different for each transcription factor, has its own input, like SOX2, yes, it will bind here, but NANOG also, even though it doesn't actually bind this OXOX motif, it actually binds here, but its binding is determined on other motifs. So you immediately see the cooperativity in that individual region. And this is kind of the difference. Like now we're collecting this information from all these different genomic regions and have to ask, okay, what is the motif? So the motif is not what we're inputting, or what also doesn't come out automatically, but we have to, we can collect those patterns and assemble it. And these are sort of network motifs. So CWM, I'm going to come back to that a little bit. So instead of just frequency matrices, where you just sort of add up the frequencies, this is actually the motif of how the network learned of how binding is driven in importance. So once we have that, we can then map also for each individual regions what the motifs are that are relevant in that region. And so now we have nicely mapped motifs that are really corresponding to our input data, which is the chip nexus data, but they're predicted completely from sequence. And now we can go in and perturb that system and say, let's say, what happens if I delete this motif? What happens to the binding of NANOG? And we do this by going back to the network, query it. So this was the oracle function of it. And so we can extract now the rules. And we can also do this in a synthetic way, where we're only taking two motifs, because you never know what's in the rest of the genomic background, and every region is different. But we can isolate the components. And this is also how we get the motif affinity. We're simply isolating its individual motif instances. And we're getting to ask the network, how important, how strong of a prediction do I get? So this is really powerful, this sort of isolation. And we can do this because we can do randomized DNA many, many times. If you would do an experiment, you never know what you're putting in, and you might by chance get some binding site. But since we're doing it many times, we're averaging this out. So this is very powerful. Okay, this is just an example of how this would look like. If we're doing this in silico, we're looking at two motifs and how binding changes. And this is sort of the description. If we're now asking of how transcription factors interact with each other, we see sort of these distance functions, what we call soft motif syntax. And you might see there's this very interesting periodicity, and you can read more about that. So that's NANOG binding, and we're working on that. So it's a very interesting protein that forms these very intricate protein-protein interactions. And the model didn't know that. These are 10 base pair periodicities. It really has to bind from one side of the DNA. But the model had no idea, but it learned the 10 base periodicity. And even though many, many people had worked on NANOG, nobody had observed this before. But you can see it in the data. You just have to know that that's the pattern. And the network saw it, so we were able to validate that. We also did some CRISPR validations. This is just to give you a little bit of a taste of that. So here's and the NANOG motif wasn't even known, I should say. Different papers were finding different things, and mostly they saw the OXOX motif. But we saw it very, very clearly, and we saw this periodicity was really jumping out of the data. And so we're able to CRISPR this and show indeed, here's just predicted and observed. And we could really see that in an individual level, we can make very accurate predictions of what happens if you mutate only two bases, right? And that's the power of that. We ask the network, okay, what if we mutate this? And of course, we do the experiment only if we see like, oh yeah, that works. If I mutate only two bases, it's going to eliminate the binding site. And that's what we did. And you can see even effects that are more long range. So you can read up on that. So that's why I'm not going to go into much detail, because I wanted to tell you something that's unpublished. So this is sort of the current work that we're doing and writing up. And this goes back to a question that I pursued, in fact, as a PhD student. And I think it's sort of a little bit on the neglected side because it's in between fields. So we know from molecular genetics that signaling pathways are key for cellular decisions, right? For cell-free decisions. So usually when you do screens for a specific phenotype, you get lots of signaling pathway components. And in fact, many signaling pathways were identified in that way intracellular because they had the same phenotype. And in addition to transcription factors, of course, right? But when you look in the transcription field, when you do genomics, Generally, people don't really care whether it's downstream of signaling or not, right? So as long as they have a motif, and the many transcription factors, and transcription factors tend to have transactivation domains if they're activating. So it doesn't seem like you really need signaling. And so transcription factor people don't really look at signaling so much, right? And there's really a hole, because signaling people usually, their studies end when it comes to the nucleus. They might look at quantitative and crosstalk and things like that. But once it's in a nucleus, it's way too complicated. And I know from my experience as a graduate student that it was, once you try to screen something downstream of transcription factors, it got way too complex and the screens didn't make any sense. So really what's happening, once you have a signaling pathway, you sort of have to integrate a lot of information, and it's very cell type specific, that then leads to a readout in that specific cell. And so to me, this was a perfect opportunity to ask that question. Okay, now we have a very sophisticated method to look at combinatorial input. Can we now better understand how signaling integrates so much information at the level of DNA? And so talented graduate student in the lab, Chiadi, kind of took this challenge. And we decided to look at the hippo signaling pathway. I mean, in part for, it's just something, this was a pathway that shipped really well, but also we felt it was very representative of how signaling generally works, because usually you have a downstream component that's already in the nucleus before the signaling pathway becomes active. And then you have a component that upon activation also binds and then activates. So that is actually very typical. And also it allowed us to look at, so we looked at embryonic stem cells before, and we wanted to show proof of principle that we can apply this technique also to a less studied cell type. And so we studied mouse trophoblast stem cells, which is sort of an early cell fate decision. Sorry. And so we all knew some of the transcription factors, of course, that were important for the cell type identity. And so Chiadi started to chip all of these. And so we wanted to take the BPNet approach to ask, what are the sequence rules by which this pathway confers activation? I mean, assume that it mediates activation. And that's also something we wanted to get at, of course. We wanted to not just study binding, but wanted to get a way of looking at activation. And so I'm going to give you just a brief overview of what we found, so you get a sense of where we're going with this. So she did the same thing. So we're doing chip nexus experiments, all these transcription factors in mouse embryonic stem cells. We're training the network to learn all these data, and then we're extracting the rules, the sequence rules that were learned by the model. And so she found, we were focusing on two examples that we found was really interesting and representative. So first of all, we see again an example of the soft motif syntax, where the distance matters in a specific way. And then we have also found an example of where the distance is very strict and is also really interesting. And we did so, we wanted to do lots of validation, because now people are still suspicious, like, well, how do you know the network learned the right thing? So initially we used embryonic stem cells, so we already knew a lot of things. So in this case, we really wanted to do lots of validation. So the way we do this is a lot of the stuff is happening with the model itself. So there are different ways of extracting the rules, like whether you do it with randomized and a genomic background. And so we usually, use many techniques to make sure that we're extracting the rules correctly. Then we're also going back to the original data and making sure that the patterns are indeed there, and not only in the original data, but also in other types of data related. Then we go back to the original cells and we do experiments to validate the rules. And in this case, we even went in vivo, although I don't have all the data yet, but it looks really promising. We can go back to the in vivo system and see that we can indeed manipulate enhancers. And in this case, we're even able to do molecular dynamic simulations. And so what we learned, we could in fact apply and could observe at the biophysical level. Okay. So again, the same kind of principle, we started off training the data at base resolution. And so this is just one example enhances for, again, for all our transcription factors. And this is the observed and the predicted. So it works very nicely, just as before. And you can even see, I think one of the strengths is that if you have low coverage, the model is really good at sort of compensating for that because it takes a rule from everywhere. And it often has really, really nice prediction for things that are not so good experimentally. That's kind of why it performs so well in some ways. And here's just a way of, after we map the motifs, we can also see that it maps it very nicely over the footprints, exactly like you would expect. Now, this is sort of just showing the motifs that we discovered for the transcription factors that we chipped. And so this is the difference between, this would be a frequency matrix, a more traditional representation, and this is the contribution weight matrix. So this is based on what the network thinks is important. In most cases, it's really similar. And then this is the advantage of the ChipNexus data, is that we can directly see the footprint over the motif. So if it's really sharp with this kind of footprint-like behavior, we know that the transcription factor binds directly. Yeah. And one thing I wanted to focus on is the TEAD4 motif. So traditionally, when people do this genomic experiment, you see the TEAD4 motif, and we did rediscover it as expected, but we also saw a double motif, and we're like, oh, that's interesting. We do see still that very spiky footprint. And we also see that YAP1 specifically binds exactly to its TET4 motif. Now, of course, that was sort of expected, but we didn't know exactly how it would turn out. But we do see that YAP1 indeed binds to where it should bind, to the TEAD4 motif. Okay. Now, we wanted to see whether YAP1 binding is the one that brings an activation. Yeah.
So the PWMs are the known motifs, or are they the
no, they're inferred from the network, but simply by looking, so we're mapping the motifs and then we're summarizing the frequencies of that. That's all there is. This is just summarized in a more traditional way, whereas the other one is summarizing the weights from the network.
But the motifs are being inferred from
so they're using TF Modisco. So basically it's, so we're extracting the regions that are deemed to be important. So the attribution, so the deep lift or SHAP. And then it takes all the regions that are important and is looking for patterns essentially. So it's clustering that. So this is sort of the downstream tools that I went in very briefly, but we can talk more about that. Yeah. So, and then it clusters that into motifs, TF Modisco. And then we can summarize that either more traditionally or in the weighted sort of the network weighted way. Usually it's very similar. Sometimes you see interesting differences. Yeah. Okay, so then we wanted to know, okay, does YAP1 bring in the activation, right? That was sort of why we wanted to study signaling. And this is just a simple way. So these are the markers for enhancer activity, I should say. So enhancer acetylation, I think I mentioned at the very beginning of the arch that acetylation is a good marker for enhancer activity, or Pol2 transcription directly at the enhancer, or the nascent RNA also directly at the enhancer transcription. And so we generated data sets, and now we're just doing a simple correlation, and we can already see that YAP1 is a factor whose binding correlates best with these markers of transcription. So that was nice to see. You can see TFAP2C also correlates quite nicely, better than TEAD4, for example. And so when we looked in, and so we're looking at regions that have really high YAP1 binding. Of course, it binds to TEAD4. We already knew that. We see very nicely the patterns of active enhancers. So this is typical of sort of on the flanks, you see the acetylation, you see Pol2 also bidirectionally transcribing, and you see the enhancer RNA. Okay, and if you look at regions that have TEAD4 but low YAP1, we do not see that pattern. So this looks really nice. Indeed, YAP1 brings in the signal. Okay, and now we want to go in a little bit more detail. So what about motif combinations? In a simple way, and my people in the lab really like it because they can do this now in R, they don't have to use the network. So we're extracting the motifs, and then what we do called island analysis. So we're simply mapping where regions where different combination of motifs are together in a certain distance. I mean, close enough that there would be an enhancer essentially. And so once we're correlating that, so this is simply looking at correlations here, we can already see that the combination of TEAD4 and TF-APA2C gives us really correlates a very high transcription here, again, with the same markers. TF-APA2C alone also is pretty strong. So that seems to be the strongest combination. And so just as a sort of test of that, so we can do a simple luciferase assay in the same cells, in the trophoblast cells, but now we're looking at activity, report activity. And so if we take an enhancer that really looks like an active enhancer, and indeed it drives luciferase activity really nicely in the cells, if we mutate either motif, there's a really strong reduction. If we mutate both, it goes even down more. But you can see that each one alone only gives you a little bit, but together, it's very strongly driving transcription. So there's clearly a synergy between these two motifs. And so now we wanted to know, okay, how is that? What exactly are the rules of cooperativity here? And so this is what is, again, we're looking at always from different viewpoints. One way is to look directly at the contribution score. So this is the initial attribution method. And we look at regions that have both the TEAD4 and the TF-APA2C motif. And so here it's aligned here to the TEAD4 motif, and we can see the model thinks for YAP1 binding, and not just needs TEAD4, but there is a contribution from TF-APA2C. So TF-APA2C also predicts YAP1 binding. So very clearly, when we look at the motifs, there are preferentially, the map motifs that we identified are preferentially very close to each other. So this suggests that there is indeed a preference that when these motifs are closer, they are more synergistic. And here we did the same sort of in silico analysis that I showed you before, where we take the two motifs in silico, and now we're changing the distance, and we're asking how strong does that to drive the binding, enhance the binding on this motif. And so what we see is that both TEAD4 and YAP1, they really like when TF-APA2C gets closer. So the closer it is, the more enhancements of binding we observe. And in fact, it's very similar, whereas the other way around is not the case. So TF-APA2C doesn't really care whether TEAD4 is there. So that suggests that TF-APA2C does drive the binding, presumably by helping TEAD4 to bind. Now YAP1 can also bind more strongly. And this is, of course, what we call the soft motif syntax, the same kind of pattern, a distance dependence that we here see. And I should say we've looked at that. It does look like chromatin accessibility is definitely one of the ways by which that is mediated. Okay, so now, of course, again, I told you we're going to do lots of validations. We wanted to test that, really show that the distance does indeed matter. And I should say there are lots of papers out there still saying that the distance does not matter. So we're not being crazy here. And so the nice thing, again, is we can ask the network, what happens if we now place the TEAD4 binding motif further away? And we do this very carefully with a minimal amount of mutations. And we can see it still binds when it's further away, but it binds less already based on the predictions. And now when we use the luciferase assay, we do indeed see quite a reduced activity of a luciferase assay in here. And I always tell Kian, yeah, but that's easy, right? It's so easy it's easy to destroy an enhancer. But she's also done the opposite of decreasing the distance and seeing an enhancements of binding, I should say. And she even did the CRISPRing here for this region. And again, this is now the endogenous loci in these trophoblast cells. And we do see a reduction of the K27 marker, and POL2 within the enhancer. So really that distance does matter. Okay, so what about the other part, the TEAD4 double motif? I think that's the really interesting part, something that we didn't anticipate. So I already showed you there were two motifs out there. The normal single motif that's everybody discovers, but there's also a double motif. And I should say we're not the first ones to see that. So like there's a Drosophila paper, characterizes really nicely, has gel shifts in there. There's also a cancer paper that describes it. So it is out there, but usually when you look genome-wide, it's not discovered. And so here's why. So when you look very closely, you can see, first of all, these are tons. These are, it was a 23,000 patterns. It's very common. And most of these sites very closely matching the consensus. And we look the most, so this is the frequency. So this is sorted by the affinity. This was the neural network trick that we can do. And we can see that the predicted to be the most high affinity are also the most frequent, suggesting they're sort of optimized for that. The cell likes that and selects for these high affinity binding sites. But for the double motif, that is not the case. So first of all, there are much fewer. And when you look at the frequency, it seems like the one that the network thinks is the strongest, it's actually not that common. And in fact, you see kind of down here, the most frequent ones are actually very low affinity. And that also explains why people usually don't find this very easy to, let's say, mean. So it looks like these motifs are often a really low affinity. And so now we wanted to see whether what do these kind of double motifs really do? So here we're taking a region with a single TEAD4 motif, and we're now making it a double motif. And we're making it either a high affinity motif or really low affinity. And this is what the neural network says. It says like, okay, well, it definitely likes that. A double motif gives very strong binding. But in this case, it's saying the double motif doesn't really bind that strongly. But when we go into luciferase assay, we can see, okay, yeah, if we have a double motif, drives activity very strongly, but even the weak double motif still drives better activity than the single motif here. So clearly they're very, very strong. And so here's an example where Chiari went and now CRISPRed this into the cells. We wanted to see what happens if we do a gain of function with that double motif. So we're getting really strong binding here. And we're now taking this in vivo. And indeed, we can now see that the enhancer, and this is a very highly expressed enhancer, or I mean, highly active enhancer based on the marks, because we wanted to make sure we can measure something. But we can drive it even stronger with this kind of very subtle mutation into a double motif. And we can even, and actually interesting, Paul is an interesting example where this did not match. And we do think that that's real in this case. In this case, it doesn't correlate with activity. But we do see the target genes, the RNA of the target genes, indeed also upregulated. And Chiari even took this. This is the example of the cells, the CRISPR cells that she's even taking now into the embryo to really show that the EZR activity is indeed increased into the embryo. We're even hoping to get a phenotype, but that's probably too much to ask for. But we want to see how much we can push this and really we're really redesigning the enhancer in a different way and see what happens now in vivo. So what exactly is this? What's happening? Why is this so strong? Now, if you look at the footprint, you can see this is, again, the nexus data, very precise. And what we're seeing here, so usually this is the protection on the left, the protection on the right. And the double motif looks like that. So it looks like you're simply having two motifs that are next to each other. But the outer sides are particularly strong, which we know, but we're working with chip nexus data means that they bind together. So it does look like they bind together, which assumes like they bind cooperatively to DNA. And that's how they mediate stronger activity. And so with luck, I have a computational postdoc who did his PhD in molecular dynamics. And we also were lucky enough that the T4 structure is already solved. So we're able to put basically now two TEAD4 on this double motif. So this was not the structure was only for one, for one side, of course. And so now we can see, well, first of all, does it bind? And we were surprised how well that fitted. And you can even see some interaction here between these two TEAD4. And so it seems really, really fitting. And so, of course, we can quantify that. So this is shown in here. So what we can quantify now is how these two sides bind to the proteins on each side. And we can also measure how these two proteins interact with each other. So that's shown in here. So what you see here, I'm not the expert on this, but you can see we're measuring the interaction area, the buried surface area. And so the more you are on the right, the stronger the interactions. And so we can see here that these two TEAD4 interacting. In fact, one is interacting stronger than the other, which is interesting. And then we also see protein-protein interactions, and they are not rock solid. I mean, they're going on and off, and a lot of times we don't see any interactions. And to me, that's actually very fitting. And when you look at the original transcriptional data, you know, the structural studies, it's consistent with that. So they're interactions, but they're not rock solid. And that's probably really good. It makes it a much more flexible system. Now we can also disturb the system and say, okay, now we're introducing a one base pair deletion into the system, which the network predicts should not be good for the binding. And indeed, when we do that, we do indeed see that now things are thrown off and the protein-protein interactions are quite diminished here. So that's interesting. So that's really, really fitting with what the network tells us. And this was sort of getting to the network tells us very precise rules. And in this case, we were able to match this even on the sort of molecular level using molecular dynamics. And not only that, but we also we can do the opposite. We said like, oh, you know, we're seeing stronger interactions here on the left side of the DNA, right? So is the left more bound than the right side? So we can now go back to the network and say, well, is this true? Do you predict that? And so this was interesting to see that indeed this was the case. So here the network predicts, this is just a summed up signal. So if we delete both sides, obviously we get nothing. But if we mutate, and this is very subtle mutation, but we're eliminating the activity. This is what happens if you delete the left side, and this is what happens when you delete the right side. So clearly the left side is stronger because you do see stronger binding here. Now, if we take that again into the luciferase assay, that is exactly matching it. So it's not just a network telling us, but it's also it's affecting the activity exactly in the way we predicted it. So again, we see here the left side alone with the right side mutated still gives us a little bit stronger activity. So I thought it was pretty amazing. They're going all the way from the biophysics to the activity in this model. And the model, the neural network is really accurately capturing this. Now we can do this, of course, genome-wide and ask, okay, is there cooperativity for all of the double motifs? The way we do this is we're predicting the binding to the double motif, and then we're asking what would each side alone do? And now we're summing this up and say, this is what it would be if it would be additive as opposed to together. And we can see indeed that there is very strong cooperativity across all of the TEAD4 double motifs. So this is what it really looks like. And this is what it would have been just by additive. And of course, these are just predictions. We can do the same thing also in embryonic stem cells where YAP1 is not in the nucleus. So we wanted to know, is TEAD4 always doing that? And the answer is yes, there's definitely cooperativity even in the absence of YAP1. So same thing. It's a little smaller. You can see the scale is less and there's also less regions, but we're still seeing the same patterns. And we can also now go in and say, well, so what were the residues by which these TEAD4 are interacting? Is this a conserved mechanism? Is this everywhere? And the answer is yes. So when we look at these residues, they are conserved from mouse to fly and humans. And we've also looked in ENCODE data. If you build the neural network models, we do see the double motifs in other cell types. So what we're thinking, and we also see that for all the TEADs, not just the TEAD4. So what we're thinking is happening is not just this one specific thing in mouse embryonic stem cells, but is really a general property of that pathway. And so with that, so the conclusion is, yes, the signaling is really precisely regulated. And we're seeing this very precise combinatorial input. We specifically looked at TFAP2C because it had a very strong input, but we also see AP1 and other transcription factors contributing. So they're very defined rules by which they interact. And we can also see that this double motif is really, really strong. And that even though it's harder to detect, we see it contributing very strongly to the signaling. And so the nice thing is that that really shows we can go all the way to the biophysics to validate these neural network models. So of course, the hope is that we can now approach this problem. You know, we can learn many different data sets and put this together in a coherent way. And that's what I was talking at the beginning. So we can now get it to sequence rules that are predictive for these genomic data sets. And so we can hopefully also link this to mechanisms. So our approach, of course, is to really go both computational and experimentally. So we're doing things iteratively, letting the network learn the rules, but then we're also going in and testing this so that we really know that we're on the right path. So the goal, of course, is to learn general rules. The problem of the cis-regulatory code is that it's very cell type specific, and that's great. You know, for specific cells, we really can do a great job, but ideally we want to do something that is generalizable. So that's kind of the big goal for the future, and we want to see which of those rules are general and which ones are really cell type specific. So for that, we need to do more, of course, but the hope is that we can build more general predictive frameworks for the cis-regulatory code to leverage all the data sets that we have. So what I sort of foresee is that we're seeing another leap in experimental biology that's driven really by computation. So an example is navigation, right? Navigation originally was very much sort of, you know, ad hoc. Then once we had educated guess, same in, you know, in biology back in the old days. Once we had genome information, or, you know, in this case, maps, it was much easier to do experiments. And we're already in this area where we have lots of information. But we also know with the advent of smartphones, it's really much easier to navigate. And so in analogy with that, once you have neural network models, it's really easier to do experiments. You can already do the experiment in silico, make predictions, and then pick the experiments you really want to do. So you don't have to do everything large scale. I mean, maybe medium scale, but it doesn't have to be always large scale. You can kind of do it in a smarter way. And I think that's really fun and exciting. So that was the end of that. And this is my lab, and I'm very grateful for a lab that has really embraced both the computational side and the experimental side. So this project I presented was Chiari's PhD work. And was really greatly helped by Charles in the lab, and then also Melanie, my bioinformatician, was really brought the deep learning from Anschluss to my lab and is instrumental in this. Of course, we have really great facilities at the Stowers Institute that makes this also possible, especially the mouse work. You know, I'm actually traditionally a Drosophila person. Didn't talk about that side today. We can also apply this to Drosophila, by the way. But mouse work is possible at Stowers. And of course, my collaborators. I'm also really grateful for tremendous ongoing collaborations. Yeah, and with that, I'll take questions.
Great talk. Couple of questions. So, do you think that if there is does that usually mean that it's a proteins are either binding as heterozygotes, for example, Box, or does STEAD4 at some low efficiency actually move and res as well? Is that actually what I'm asking is if there are some soft distances and some strict distances. When it's a strict distance, doesn't mean it's an obligate hetero or homodimer, whereas when it's more soft, is it binding as a single protein as a homodimer?
Yeah, I think you're right. And so to me, it looks like the strict syntax is really mediating protein-protein interaction from what I can see. I haven't seen a good example where that wasn't the case. So in the embryonic stem cells, there is a lot of strict syntax that people publish, but it's coming from transposable elements. So it's sort of artificial. It's not actually a strict requirement. It's just present many, many times in that distance, but that's artificial. And so whenever that's actually relevant, it seems like it's protein-protein interactions. The bigger question is therefore, what if it's not, right? And so the soft syntax can be mediated by different mechanisms. And we do see in the original paper, for example, SOX2 and NANOG, they do interact by protein-protein interaction, but also have a soft syntax. And because they're mediated by protein-protein interactions, it could be that at close distance, this is indeed driven by that. But at longer distance, it seems more likely driven by chromatin. And we've done more work on that, which I didn't present. It really does seem to be a big thing. So usually when you see a function that goes into the distance and is still quite enhanced, it's usually nucleosome distance, and it's driven by chromatin accessibility. So that's something that we're really following up in quite some detail. But that doesn't mean that it's the only mechanism. So there's still other possibilities. So other mechanisms have been described where protein can interact, not by protein-protein interaction, but simply, I don't know, it's called vibrational, I don't really know, but sort of where transcription factor binds, but leaves something in the DNA that allows the second transcription factors to bind better. So there are ways there could be, or could also be histone modification. So there could be other mechanisms too that could, of course, that's the thing we don't see, right? And we have to test that sort of more experimentally to see, like, hey, does this describe, in this case, this function? And we haven't explored all the possible mechanisms, right? So, but the protein-protein interaction definitely is a big part.
And then in your double-teed motif, we generally see that the vicinity of the double-teed motif, is it depleted for the TFAP2C motif? Because what I'm wondering is that you need the double-teed when there's no TFAP2C nearby to help it.
Interesting. I mean, clearly it cell takes both, right? Sure. But it's a good question. I don't think we've looked at that specifically, because you could argue that if there's a double motif, maybe you don't want a really strong motif. Maybe it's too strong for some reason. And in fact, the fact that Pol II no longer can transcribe well there, maybe there's something to that. But I don't know whether it would be too much if TFAP2C were there. Actually, we have not looked at that. If you reach a threshold which it's too good, it's too strong, I don't know. We would predict that it would drive activity even better if you have a double motif and then TFAP2C, but you haven't specifically tested that. So, yeah.
Do you take into account the chromatin status while like taking data for training and testing?
No, we don't. So we train on 1 kb regions, and so there could come, and so we're training on regions that are bound. So by definition, they are accessible. We're not including completely closed regions. And I think that would be hard for the model to learn, because if it is sort of a long-range repression, so if you take a region out of its context, it might drive activity, right? And the network doesn't know that, what's kind of on the side, right? So we're really training it on regions that have the possibility of binding, because we know they're bound, right? But the accessibility could be different depending, and we know this when we're integrating key elements into the Drosophila genome. It really depends on where it lands, of how strongly reporter activity is driven. And this part of the chromatin, we don't know, right? So the model doesn't really learn that perfectly. And we do see that it learns the profile better than the counts. And this is because of that, because we're taking it out of the chromatin context, and it can't learn this information from elsewhere. And so overall, it still does good, but not fantastic on the count side, because it doesn't have that chromatin information. But that's why we're leveraging the profile. So the idea is that the profile won't change, whether it's in a good chromatin, very, accessible chromatin, or, more suppressive chromatin, the profile is still the same. And that's why the model performs so well.
So right now, the way we've been applying these models is training one convolutional neural net at a time, and then you're post-processing it as a interpretation of the model. We are trying to figure out whether there are rules. I was wondering if it is possible to train the model on 4, like how many ever chiplex data sets you have, and whether that might be other rules if you are simultaneously modeling.
Yeah, it's a really good question, and it is very discussed in the field. I don't know. So we did actually do a multitask training in the original paper, and also, but in fact, Anshul is always saying like, no, you do single models, and we have done single models, and we do get the same results. but there is that, and in our case, it's so clean and the data is so precise that it really doesn't make a difference. But we have seen cases where it does make a difference. And you can imagine that if it has learned a motif from another task, it will try to apply it to learn this task. And so that can be good or bad. And this is where the debate is. So it can be good if it's actually biological information that is appropriate to learn, then gets a nudge to learn this information. But it can also be bad because if you train, for example, multiple cell types simultaneously, it will get information that's really not relevant for that cell type because that transcription factor is not there to recognize that motif. But it may still use that motif to say, oh, if that's there, then that's probably negative, that's not good. And it does use that. So that's sort of the downside of that. Like, do you trust that if you do? And that's also the question of kind of, it's a debate in the field. It's not just like our 4 data set, but if you take every ENCODE data set, like Informer model, you train everything at once. So the idea is, of course, that it should do great. It should learn general rules. But the truth is it doesn't quite work, unfortunately. It also learns artifacts. And I mean, this is a big debate. Is this the right approach? Or if this is the right approach, how do we do this properly that it doesn't learn these artifacts? So yeah, it's it's a good question.
Are the questions online?
How do I go?
Oh, there's a chat.
Oh yeah.
Okay.
Can I move this? Yep. Hi, Julia. Okay.
Oh,
From John Kuliak. Is there a way to access presentation recording? Oh, I see. I'm sorry.
eventually,
Yeah.
yes, it will be available online.
Okay. Eventually. So sorry.
They're all available online.
It will be posted on CSI.
Uh-huh.
I sent them the links regularly.
Mm-hmm. Okay.
One other question. I know we are
Yeah.
ending.
Huh?
So chip nexus is a very useful data set, but it's not there widely available.
Yeah.
Is it possible to predict it? I mean, from sequence, maybe the same thing, but is it possible to predict chip nexus using other modalities or maybe the low resolution chip-seq data?
Yes.
And
So we did show in the original paper, and Anshul has done that also, so that you can do the same learning paradigm on chip-seq data surprisingly well. Like our motivation originally was to understand what's all in this sort of very precise data sets. But it turns out a lot of the resolution and the fine-grained information can also be obtained by chip-seq data. So the motifs are actually the same. What is not quite the same is the resolution. So sometimes if you have 2 motifs that are very close, in chip-seq data, you won't be able to resolve that. Also, you won't be able to see what's directly bound. Like we see this very sharp footprint, so we know, okay, that's where the transcription factor sits directly. In chip-seq, you don't know that. You only know that that motif is contributing, but you don't know in what way, because you can't read it out. So there are some things that are different, but overall, it turned out to be surprisingly powerful. Yeah. So there is actually more information in the shape than you would expect. That's kind of the good thing.
okay, so if there are no more questions, let's thank you.
Mhm.
Amazing talk.